Bioluminescence is the natural phenomenon of light emission by living organisms, an ability possessed mainly by marine organisms, including some dinoflagellates. Marine bioluminescence is most often seen as a blue color, tuned to the wavelengths traveling furthest through the water. Dinoflagellates are the main eukaryotic protists that have this ability. Bioluminescence is estimated to have evolved independently over 40 times, producing a diverse range of chemistries behind bioluminescent reactions. In dinoflagellates, light production occurs in a certain organelle called a scintillin. This contains luciferin substrate, the luciferase enzyme, and in some dinoflagellates, a luciferin binding protein. To begin, a force is exerted on the outer cell membrane, which is translated from a mechanical signal to an electrical signal on the vacuole membrane. To do so, calcium concentrations in the cytoplasm increase, mainly from internal sources. This depolarizes the vacuole membrane, allowing a scintillin to connect to it. As a result, the action potential can travel across the scintillin, activating voltage-gated proton channels. The influx of protons from the vacuole lowers the pH of the scintillins and activates their bioluminescent chemistry. Bioluminescence serves many important roles, including attracting mates and luring prey. However, all the functions of bioluminescence in dinoflagellates are still not completely understood. One theory of bioluminescent function in dinoflagellates is the burglar alarm theory. This stems from the idea that most common grazers of dinoflagellates, the copepods, are often too small to elicit a high level of bioluminescence. Instead, the behavioral response of a copepod grazer to the flashing of a dinoflagellate, rather than the flashing itself, attracts the attention of the copepod's predators that rely on flow sensing for predation. This behavior works even with the small grazers that only cause single cells to flash.
So, what is the evidence for this theory? To gather evidence for the theory, there were quite a few papers that tested the impact of bioluminescence on dinoflagellate consumers. Here we have gathered three of them with visual data and a fourth that does not have any visual data. Mensinger and Case in 1992 studied mysids and midshipman fish alongside dinoflagellates. In Figure 1 on the left, there is a positive correlation between the average number of mysids consumed by fish and dinoflagellate luminescence up to a point, around 17.5 concentration in cells per milliliter. In Figure 3, the predation attempts by the fish are initially higher with bioluminescence and then decrease. This study showed that the burglar alarm theory worked until the density of dinoflagellates inhibited successful detection of prey. A later study by two different scientists, Abrahams and Townsend, had no visual data. Flesher and Case in 1995 had another test of the theory with cephalopods and lower trophic levels, including mysid, grass shrimp, and mosquito fish. The study not only showed evidence for the burglar alarm hypothesis, but also showed that apex predators are able to capitalize on bioluminescence in otherwise dark settings. They were even able to detect eye movement of the cuttlefish tracking the bioluminescence. In Figure 3, Flesher and Case showed cephalopod predation on mysid with no inhibition at high densities. Scotophase is a time period in which dinoflagellates are using bioluminescence due to dark lighting conditions. This is shown in the darker bars. The different lighting times were achieved by adjusting the lighting cues of the two populations to have alternating bioluminescence. Figure 8 on the right illustrates the strike frequencies of attacks by cephalopods on grass shrimp with no evidence of an optimal density. In both cases, there was a positive correlation between bioluminescence and successful consumption of dinoflagellate predators. The final study, Wong et al. In 2023, revisited the burglar alarm theory with copepods to examine the hunting behavior of dinoflagellate predators and its association with flashing. Their test subjects were copepod nauplii and copepod adults. Copepod nauplii were the predators of dinoflagellates in this case. In Figure 2 on the left, the ingestion and clearance rates of nauplii regarding dinoflagellates were significantly higher in non-flashing environments. Non-flashing refers to a lack of bioluminescence, while flashing is with bioluminescence. In Figure 4 on the right, only the ingestion rates of adult copepods on nauplii were significantly higher in flashing environments. The study also included nuclei jump frequency, velocity, and duration during dinoflagellate foraging to see if behavior of the predators affected the foraging pattern of the adults. Nuclei jump frequency was consistently significant relative to dinoflagellate concentration across 3 dinoflagellate species.
The 3 studies which provided evidence in support of the burglar alarm hypothesis varied in reliability. Minsinger and Case's study, for example, was unable to identify the specific cause for decreased predation at higher dinoflagellate concentrations. Fleischer and Case's study, however, observed conflicting evidence. No significant decrease in predation at similar concentrations. Several factors could have contributed to this. For example, Fleischer and Case used 12-liter aquaria for cuttlefish, and Minsinger and Case used a range of much smaller containers for teleosts, in which high luminescence might more easily hinder foraging efficiency. Both studies only had one individual predator per container, and similarly sized predators. It may be worth investigating the effect of spatial boundaries on this observation. In addition, while both are visual predators, cuttlefish possess polarized and light-filtering vision. Fleischer and Case were able to capture cuttlefish eye movement while it tracked its prey. This feature may have contributed to the results observed. Abraham's and Townsend's study, unlike those performed by Mensinger and Case or Fleischer and Case, did not use increasing concentrations of dinoflagellate cells. Instead, they measured copepod mortality rate in containers with dinoflagellates only during their luminescent or non-luminescent phases. They did not record the experiment or actively observe species interactions, partially due to contamination. Ultimately, they only showed that a visual predator was able to hunt better in a more well-lit environment, rather than due to the burglar alarm mechanism. Huang et al. Suggest an alternative mechanism to that proposed by the burglar alarm hypothesis, where an individual cells benefit from having luminescent ability, rather than only larger populations of cells. They argue that, in the case of the burglar alarm mechanism, non-luminescent cells that benefit from the system without contributing should be selected for, yet this is not the case. Additionally, the burglar alarm mechanism doesn't work as well at low populations or with single cells, and a costly trait that doesn't provide an advantage should get selected out of the population, even in those that are clonal. Yet this also isn't observed.
So, how's the viability of the hypothesis? Overall, the data seems to support the burglar alarm hypothesis being a viable explanation for dinoflagellate bioluminescence. Dinoflagellate flashing generally increase the predation on dinoflagellate predators by a higher trophic level predator up until a certain dinoflagellate concentration. However, there were some limitations and uncertainty in the studies, largely stemming from differences between a lab setting and natural conditions. One aspect of the hypothesis that was touched on earlier is how dinoflagellate luminescence is more beneficial to the population than to a flashing individual. So what, are flashing dinoflagellates sacrificing themselves for the good of the species? Well, no. While the burglar alarm mechanism may result in fewer dinoflagellate deaths, selection depends on the survival of individuals, so this would theoretically make the burglar alarm selectively unfavorable. But Abrahams and Townsend argue that local populations of dinoflagellates are often made up of clones, so even though flashing individuals may die, the survival of genetically similar individuals increases, which points towards selection favoring luminescence. However, Huang et al. Reasoned that the burglar alarm is probably not the primary reason for dinoflagellate bioluminescence, because it promotes survival of the population, not the individual. They argue that this holds true even for clonal populations. Another reason why it is likely not the primary function of luminescence is that it does not work well at low cell concentrations, as we discussed. As far as the broader implications go, the burglar alarm hypothesis is one of many possible reasons for bioluminescence and can coexist with other theories. As Abrahams and Townsend wrote, it should be noted, however, that the burglar alarm is not a mechanism that must operate independently of other proposed functions. Bioluminescence may still provide other benefits to dinoflagellates. For example, another reason for dinoflagellate luminescence is to startle predators, a defensive mechanism where flashes of light are used as a form of predator intimidation, according to Haddock et al. Figuring out the reason for bioluminescence can help us understand the selective pressures on an organism or population.
As we discussed earlier in the course, over 80% of animals in the pelagic zone exhibit bioluminescence in some way, so it is an important phenomenon to understand. Patterns of bioluminescence can help scientists understand the interactions among individuals, populations, and communities in our beautiful ocean. Most proposed reasons for bioluminescence involve interactions between organisms, including predator-prey interactions, communication, and mating.